morning friends. Today we are going to start with second chapter of 12th standard, that is sexual reproduction in flowering plants. Right, so we are talking about flowering plants. So we are very clear that we are talking about angiospermic plants. Gymnospermic plants possesses naked seeds. They are non-flowering plants. Right, so talking about the structure of a flower, right, a flower is a modified stem which rests on a body. That body is called as a thalamus or receptacle. Right, so this is thalamus or receptacle. Which are the parts of the flower? In the center there is pistil. Pistil is a part of female reproductive system of flower. It consists of three parts: a bulb-like structure which is called as stigma, then a long, elongated, narrow style, and a swollen structure which is called as ovary. Right, so these parts together form a pistil. Right, so we will name it. This is a bulb-like structure which is called as stigma. This is an elongated structure called as style. This is a swollen structure which is resting on receptacle which is called as ovary. And these three structures together form pistil, and pistil is a part of female reproductive system. The second part of the flower is stamen. Right, so we will draw a few stamens. These are the stamen. It consists of a long slender structure again which is called as filament, and this are the lobe-like structures which are called as anther. This anther and filament together form stamen, which is a part of male reproductive system. Male reproductive system is also called as androecium. Female reproductive system is also called as gynoecium. Then the next part of the flower is beautifully colored whorls. These whorls are called as petals, and all the petals are together called as corolla. Okay. Beyond it is again one more whorl which is called as sepal, and all the sepals are together called as calyx. So this is the structure of a flower. A flower rests on a thalamus called as receptacle. In the middle part of it is a female reproductive system which is called as pistil. It consists of a bulbous stigma, an elongated style, and a swollen oval. On the either side of it are the parts of male reproductive system, that is androecium. Those are called as stamen. Stamen consists of two parts. Right, this lobe-like structure is called as anther, and this long slender structure is filament. Then the next whorl is petals. These petals all together are called as corolla, and the sepals all together are called as calyx. These walls, that is calyx and corolla, are called as accessory walls. And this androecium and gynoecium are called as the main required walls. So this is the structure of flower. So the next topic is structure of stamen. So we know that stamen is a part of male reproductive system. All the stamens together form androecium, that is male reproductive system. So which are the parts of stamen? Stamen consists of two parts, that is anther and filament. Right? So this is anther and this is a long slender filament. And this filament is attached to a base that is called as a thalamus or a receptacle. Right? So this structure is stamen. It consists of anther and this is filament. And the part of the filament which is attached with the anther, this part of the filament is called as the distal end of the filament, and this part of the filament which is attached to the thalamus or receptacle is called as the proximal end of the filament. Now this anther, the anther can be of two types. It can be made up of a single lobe, which is called as monothecous anther, or it can be made up of two lobes. Such type of anther is called as dithecous anther. So monothecous anther and dithecous anther. Here theca means lobes of the anther. So anther with a single lobe is called as monothecous anther, and anther with two lobes are called as dithecous anther. Example of monothecous anther is the anthers which belong to Malvaceae family, and most of the anthers which are found in the angiospermic plants are of dithecous type. The diagram which we have drawn is also a diagram of a dithecous anther. Right? If we study the diagram of the anther, right? So these are the two thecas of the anther. In between these two thecas, there is a connective tissue. This tissue, which is connecting the two theca of the anther, is called as connective. This tissue is sterile tissue. Right? So there are two types of tissues, sterile tissue and fertile tissue. This tissue is a sterile tissue, which means this tissue does not take part in the process of reproduction. Right? So the two anther lobes are connected by a tissue that is called as connective, and that connective is a sterile tissue, which means it does not take part in the reproduction. The attachment of the anther with the filament is of different types. Which are those types of attachment? Right? So these are the lobes of anther. We are taking the common anther, that is dithecous anther. Monothecous anther is seen in very few families such as Malvaceae family. So if there are different types of attachment, if these two lobes of the anther are attached with the filament, so the base is attached, such type of attachment is called as base-fixed. If this whole part, that is connective, is attached with the filament through its dorsal side, right, then it is called as dorsifixed. If it is as adamate. And there is one more type in which the anther lobes are horizontally placed. So these are the anther lobes, and this is the attachment of the filament. Such type of attachment is called as versatile. This versatile type of attachment is the characteristic feature of anemophilous flowers. Anemophilous flowers are the flowers in which pollination takes place by wind, and in such flowers the anthers are such attached with the filament that these anthers are dangling, right? So those are not attached thoroughly with the filament, but the anthers are attached horizontally, and so those are dangling. That is why there will be easy dehiscence and release of pollen grains, right? So these are the four types of attachment of anther lobes with the filament. If it is attached through the base, it is called as basifix. If it is attached through its dorsal side with the filament, it is dorsifix. If the entire length of connective is attached with the filament, it is called as adamate type. And if the anthers are horizontal and dangling, such as in anemophilous flowers, then such type of attachment is called as versatile attachment, right? As we have studied that the most common type of anthers are dithecous anthers, and theca means lobes. So if we study the structure of this anther, then it is dithecous, which means it is made up of two thecas. So if we will see an overview of it, then it would be like this. So this is one theca, and this is the another theca. So this is one theca of the anther. This is one lobe or one theca of the anther, and these two thecas are connected by a sterile connective tissue. Right? So this is sterile connective tissue. So these are the two thecas of the anther. Each theca of the anther will possess two microsporangiums. Right? So this are called as microsporangium. Those are the sac-like structures. One anther lobe will possess two microsporangiums. Another anther lobe will also possess two more microsporangiums. So this is a bilobed anther, and it possesses in total four microsporangiums. So you can say it is bilobe and tetra. Sporangiate. Right? So this is the structure of a typical dithecous anther. Two lobes are there. The two lobes are connected by a sterile tissue which is called as connective, and each lobe possesses two microsporangiums, and these microsporangiums will be filled with the cells. These cells are actually undergoing the process of microsporogenesis. These cells are called as archisporangial cells. So this is the overview of the structure of an anther. In the next lecture, we will study about the internal structure of the anther. Our next topic is internal structure of anther. As we have studied earlier, that a typical anther is made up of two lobes. That is, it is dithecous. Each lobe will possess 2 microsporangium. So it is bilobed and it is tetrasporangiate, right? So in total 4 microsporangias are present, right? This is present in a young anther. As and when anther matures, so we know that anther is a fertile tissue of the male reproductive system. As and when anther matures, it gets bigger in size. When the anther gets bigger in size, this microsporangiums, those are the sac-like structures, those will also get bigger in size. And as the anther matures, near to its maturation, those microsporangiums are called as pollen sacs. So a mature anther will have pollen sacs, right? So internal structure of anther, the anther wall consists of 4 layers, right? So there are 4 layers which are present in anther wall. We are studying these layers from outer to inner side, right? So this is the anther wall. This is called as anther wall. This is microsporangium. So this anther wall is made up of 4 layers. Which are those 4 layers from the outer side? So the first layer is called as epidermis. This epidermis layer is made up of flat epidermal cells. It is forming the outer layer. So outer layer of anther wall is made up of flat cells. Those flat cells are called as epidermis, right? So that is the first layer, that is epidermis, which is made up of flat cells forming the outer layer, right? The second layer, right, this epidermis layer is a single layer. So it is made up of a single layer of flat cells. The second layer is called as endothecium. This endothecium is also made up of a single layer of radially arranged compact cells with callose thickenings, right? So the outer layer is epidermis. Epidermis is made up of single layer of flat cells. Inner to epidermis is a single layer of the cells which are radially and compactly arranged. That layer is called as endothecium. And in between these cells of endothecium, there will be thickening. That thickening is made up of callose. Right, so this is the second layer of the cell. This layer of the cell is called as endothecium. It is a single layer of radially arranged and compact cells, and these cells are together placed by callose. Callose is functioning as a glue material to keep all these cells together and compactly arranged. Then the third layer is called as middle layer. It consists of 3 to 4 layers of parenchymatous cells. Consists of 3 to 4 layers of parenchymatous cells, right? So again we will draw the next layer. This layer, third layer, consists of middle layer, and this middle layer is made up of 3 to 4 layers of the parenchymatous cells. So epidermis is a single layer of the cell, endothecium is also a single layer of cell, whereas the third layer, that is middle layer, consists of 3 to 4 layers of parenchymatous cells. And all these 3 layers of the anther wall, right? So these 3 layers, or we can say that these 3 layers of anther wall helps in dehiscence of anther. What is dehiscence of anther? So as I told you that a young anther is present, it is a fertile part of male reproductive system. As and when anther matures, within this sporangiums there will be formation of the male gametophyte, that is pollen grains. Once these pollen grains, mature pollen grains are formed, this anther ruptures and these pollen grains are released. That process is called as dehiscence of the anther. This dehiscence of the anther will take place from this place, right? This is the part from where dehiscence of the anther takes place. This part is called as stomium, right? It is called as stomium, where dehiscence of the anther takes place. What is it, right? So we know that anther wall, and these are the 3 layers of the anther wall. We have to study one more layer of the anther wall. So these 3 layers of the anther wall, as and when the anther matures, will become mature, right? So these 3 layers will become fibrous, and when it becomes fibrous, so it will become fibrous all along the length of the anther wall except at the place of the stomium. Stomium is the place where this will not fibrous. So what will happen? These 3 layers will become fibrous, so it will become rigid. So the entire anther wall will become rigid except at the place of stomium, where it is not going to become rigid, and so it is going to rupture, right? So this part becomes rigid and this part doesn't become rigid, so it is going to rupture. So stomium is the place where dehiscence of the anther takes place, right? Then the 4th layer is called as tapetum. It is the innermost layer of the anther wall, right? So the 4th layer disturbing. The 4th layer is called as tapetum, right? The tapetum layer consists of pyramidal shaped cells, right? So it consists of pyramidal shaped cells with a very thick and dense cytoplasm present within it, right? This is a very important layer, that is tapetum layer. It is the innermost layer of the anther wall. Right? So the innermost layer is tapetum layer. Again, it is made up of single layer of the cells. What are this type of cells? These cells are pyramidal shaped. They possess dense cytoplasm. These cells are multinucleated. Very important. These cells are multinucleated, which means a single cell will possess many nuclei. How can these cells be multinucleated? It can be multinucleated either through endomitosis, right? What is endomitosis? So there will be multiple divisions of the nuclei within a cell. That is called as endomitosis, or by the process of syncytium. What is syncytium? karyokinesis is not followed by cytokinesis. So the cells will be multinucleated cells. So the innermost layer is tapetum. Tapetum is made up of single layer of pyramidal cells. This cells possesses dense cytoplasm. Those are multinucleated, and they are multinucleated either through endomitosis or through syncytium. What are the functions of this tapetum? The first function is it provides nourishment to the developing microspores, right? So we have studied that within this microsporangium there will be the archisporangial cells, and these cells will function as microspore mother cells. Those cells are going to undergo meiotic division to form microspores. So when these cells are undergoing meiotic division, they require nourishment. A single cell is going to form 4 haploid daughter cells. So they require nourishment. That nourishment is provided by the tapetal cell. So it provides nourishment to the developing microspores. Then the second function of the tapetal cells is secretion of enzymes. Which enzyme is particularly secreted? The enzyme is Calais enzyme. In the further videos, you will study that when the pollen grains are formed, when the microspores are formed, those microspores are attached with one another by callus. Say for example, this is a single microspore mother cell. When this single microspore mother cell is going to undergo meiotic division, it is going to form 4 microspores. So say these are the 4 microspores which are formed. These microspores are first of all glued together by callus, right? But that callus will be disrupted. Those callus will be disrupted by the Calais enzyme, and that Calais enzyme is secreted by tapetum. So the function of tapetum is the secretion of Calais enzyme. Then the third function of tapetum is production of Ubishk bodies, right? And this Ubishk bodies possesses sporopollenin, right? This sporopollenin is the most resistant substance known in living world, right? So the cells of the tapetum are going to produce Ubishk bodies, and these Ubishk bodies are going to possess sporopollenin, and this sporopollenin forms the most resistant substance known in the world, right? So that is about the internal structure of the anther. The anther wall is made up of 4 layers from outside to inside. Which are those 4 layers? The outermost layer is epidermis, which is made up of single layer of flat cells. Inner to it is endothecium, which is again made up of single layer of radially arranged compact cells, which are together glued by callus thickenings. Then the next layer is middle layer, which is made up of 3 to 4 layers of parenchymatous cells. These 3 layers, epidermis, endothecium and middle layer, will help in dehiscence of the anther. What is dehiscence of the anther? It is rupturing of the anther and release of the pollen grains when the anther matures, right? And the innermost layer is the tapetal layer. This tapetal layers, the cells of the tapetal layer are having very peculiar features, pyramidal shaped. Those are having dense cytoplasm. Those are multinucleated, right? How is it multinucleated? Through endomitosis or through syncytium. What is the function of this? It provides nourishment to the developing microspores. So inside there will be archisporial cells. Those will function as microspore mother cells. This will produce microspores, and microspores are going to produce pollen grains. So it provides nourishment for it. Then secretion of the enzymes, particularly Calase enzyme. And what is the importance of this Calase enzyme? This Calase enzyme is required to dissolve callose of microspore tetrad, right? And other function is production of Ubishk bodies. This Ubishk bodies possesses sporopollenin. It is the most resistant substance known in the world, which means if we are consuming this pollen grains, this sporopollenin forms enzyme of pollen grain, and it is made up of sporopollenin. It is very resistant. If we are consuming it, still the acid, hydrochloric acid, which is present in our stomach, is also not able to resolve. So that is about the internal structure of the anther.